All right, so now we'll be starting our oral abstract session. So I'm going to invite Vanessa Schults forward. Vanessa is a senior master's student in the Department of Medical Microbiology and Infectious Diseases within the Dr. McLaren's lab studying HIV. And was selected as one of 2 oral abstract presenters today. So please join me in welcoming Vanessa.
So hello everyone. I hope you can see all my slides okay. I'm just going to pull up my laser pointer here. All right, so the title for my talk with you guys today is that HIV sequence diversity is impacted by variation at CHD1L and HLA class 1 loci in a South African population. And so first, HIV set point viral load is a predictor of disease progression and transmission. And we also know that it's variable between individuals. We know that host genetics contributes to about 25% of the variability we see in viral load between individuals, and the virus itself contributes to about 30% of the variability we see. HIV is also one of the greatest risk factors for acquiring active tuberculosis, and this is because of the important role that the cell-mediated immune response plays in the defense against active tuberculosis. And so, as I said, HIV disease progression HIV set point viral load is a predictor of HIV disease progression, which is characterized by the loss of CD4 positive T cells, which is part of the cell-mediated immune response. And so therefore, the restriction of HIV set point viral load is not only key to reducing HIV disease progression, but also the risk of acquiring active tuberculosis. And so Barth et al. Conducted a study in 2013. It was a genome-to-genome analysis where they had host genome, viral genome, and set point viral load data available. And what they found is that viral load was significantly associated with alleles in the HLA region on chromosome 6. And you can see that in just to the right here, figure A, with the HLA region highlighted here in this box. What they also found was that there were viral amino acid variants that were significantly associated with alleles in the HLA region. But what's important about these associations is that the magnitude was much greater than with set point viral load. And so you can see the comparison of those 2 associations between panels A and B here, with the amino acid variants being much stronger. And when they looked at the spread of these amino acid variants across the HIV proteome, they found that the associations were completely across the HIV proteome. And so essentially, Barth et al. Showed that the HIV genome or proteome provides more power to assess the host impact on HIV evolution. Unfortunately, the Barth et al. Study, a majority of genome-wide scans are conducted in individuals of European ancestry, meaning that these scans miss the populations that are most impacted by HIV, but also the majority of human genetic diversity. And so to address this, our lab group conducted a genome-wide association study in more than 3,800 individuals of African ancestry. And what they found was that there are variants in a locus on chromosome 1 near a gene called CHD1L that is associated with lower set point viral loads. And what's important about this association is that it's specific to individuals of African ancestry, and it's reproduced across different African populations. And I'll also just make a note that CHD1L is a helicase-like protein that has been shown to restrict HIV replication in vitro. However, some individuals that have these protective alleles on chromosome 1 still experience high viral loads. And so that trend is shown in the box plot to the right here, where you have your log 10 set point viral load on the y-axis and the allele dosage on the x-axis for this particular SNP, with AA being homozygous for the reference allele and GG being homozygous for the protective allele. And you can see that generally as individuals gain a copy of that protective allele, the viral load does go down, but there are still some individuals experiencing this high viral load, as shown by the upper quartile of this box. And so based off the Barth et al. Study and then other studies that have looked at HIV escape from HLA, we hypothesize that there's selective mutation of the viral genome that is allowing HIV to escape restriction by CHD1L. And the gap in knowledge that we're trying to address here is that the impact of this chromosome 1 locus on viral evolution has not yet been investigated. And the objective of the study was to assess the association between amino acid variants in HIV reverse transcriptase and also protease with host alleles near CHD1L and then also HLA. And so to briefly review the methods, we acquired genome-wide data for individuals part of the HIV Incidence Provincial Surveillance System, or HIPS, and then also individuals part of the Center for the AIDS Program of Research in South Africa study, or CAPRISA. This is also known as the tenofovir gel study. And it's important to note that both of these cohorts come from the province of KwaZulu-Natal in South Africa, which has the highest burden of HIV worldwide, but also the highest burden of HIV-TB co-infection. And so from this data, we selected 4 protective chromosome 1 SNPs, and then also 23 2-digit HLA class 1 alleles. We then matched these individuals to protease and reverse transcriptase sequences. I will mention that the host sequences and viral sequences had been previously generated. So we did our best to retain as many individuals as possible within the study. And so this left us with up to 148 individuals to do testing. The viral sequences were then aligned and translated to protein sequences using the Stanford HIV-DB online tool. And then with these protein sequences, we selected codons to do statistical testing using the thresholds that I have here. And that was just to ensure that we would have enough consensus and variant amino acid calls to do the testing. And then statistical testing was done in Plink, specifically using logistic regression. And for every codon, the amino acid calls were assigned a binary value. So consensus amino acids were assigned a 0, variant amino acids assigned a 1. And then associations were considered significant with a false discovery rate of less than 5%. So we first acquired the data for the HIPS cohort, and so this served as our discovery cohort. And what we found was that all 4 of the protective chromosome 1 SNPs that we selected for testing were significantly associated with an amino acid change in reverse transcriptase 248. We also found that there were 3 HLA alleles that were significantly associated with amino acid changes in reverse transcriptase. We did not have any significant changes for protease. We next acquired the data for the CAPRISA cohort to replicate the findings from the HIPS cohort. And after these were analyzed together, we found that the only association that remained significant was the one between HLA-B81 and reverse transcriptase codon 4. The next thing we wanted to look into was how viral load would be affected by host allele dosage or variant amino acid dosage as a better way to visualize viral escape. And so the first thing we found was that viral load was variable in HLA-B81 carriers and also carriers that had variant amino acids and reverse transcriptase codon 4. And so that trend is better shown in the box plots below. So for these box plots, you have your log 10 viral load on the y-axis, and then either host allele dosage or variant amino acid dosage on the x-axis. And so in the first box plot to the left, we could see that individuals with 1 copy of the HLA-B81 allele tended to have lower viral loads than individuals without. We didn't have anyone that was homozygous for it. And then when we look at the box plot to the right, so this only represents HLA-B81 carriers, but we see that HLA-B81 carriers with a variant amino acid and reverse transcriptase codon 4 tended to have higher viral loads. And so this is exactly what we would expect to see in a case of viral escape, where we have a host allele that is restrictive on HIV, HIV then mutates, and in this case, that mutation is advantageous, and we're seeing high viral loads. The next thing that we kind of also wanted to look at was to see if there was a kind of biological mechanism that was working here. And so I ran a epitope prediction using the IEDB TIPI Tools online tool. And I found that this epitope, which is called SL10 and covers reverse transcriptase codon 4, was found to be restricted by HLA-B81 with an IC50 of 567 and a percentile rank of 0.04. I'll also note too that this association between HLA-B81 and reverse transcriptase codon 4 has also been previously described, but reverse transcriptase codon 4 hasn't yet been optimally defined as an MHC ligand. So the next thing I did was I looked at the exact same thing, but for chromosome 1 SNP carriers. And so if we look in the box plot to the left here, there's really no difference in viral load, whether individuals had 0, 1, 2, or all 4 of those protective chromosome 1 SNPs, which was not exactly what we had expected to see. We would have expected a lower viral load based off our previous study. And then if we look in the box plot to the right here, we can see that again, there's really no difference in viral load, whether individuals had a consensus or variant amino acid and reverse transcriptase codon 248. And so to summarize the findings for this study, we found that amino acid variants in reverse transcriptase was associated with chromosome 1 SNPs near CHD1L in the HIPS cohort. However, we didn't see any variability in viral load in those chromosome 1 SNP carriers or carriers that had variant amino acids. And unfortunately, the chromosome 1 SNP associations were not replicated within the Caprisa cohort. And then lastly, the SL10 epitope, which covers reverse transcriptase codon 4, was associated with HLA-BD1, and we also see that viral load was variable in HLA-BD1 carriers and also carriers with variant amino acids. Conclusions for this study was that despite having a smaller cohort, so again, only 148 individuals, we were able to identify the association between HLA-BD1 and reverse transcriptase codon 4, which suggests that our approach was valid in principle. However, moving forward, we want to increase the cohort size and then also access whole genome sequences for HIV to more fully investigate the CHD1L impact on HIV evolution. And then in more of a broad sense, it's imperative that similar studies are done in additional cohorts. So only 20% of genome-wide association studies are done in individuals of non-European ancestry, even less so for individuals of African ancestry. And also there's been no large-scale genome-to-genome analyses. And so moving forward, it's really important that we get more representation from these populations that are most impacted by HIV. And so just to finish things off, I'd first and foremost like to thank the Caprisa and the HIPS team for providing their data and then also providing input on my work. And then also to the Caprisa and HIPS participants. Without them, I would have no project. Thank you to my lab team for all of your support and all of the training, to my committee supervisors as well. And that brings me to the end.
Now our second presenter is Allison Balasko. Allison will be joining us online today, so you'll see her on the screen eventually. Hi, Allison. So Allison is a senior PhD student, also within the Department of Medical Microbiology and Infectious Diseases within Dr. Folk's laboratory. So take it away, Allison.
My presentation title is Function and Exhaustion of Invariant Natural Killer T Cells in People Living with HIV. And I'm in Dr. Folk's lab. There we go. So just a bit of background on my project. So I study something called immune exhaustion in chronic HIV infection. So basically this overall is a loss of immune system effectiveness. This happens despite people being on therapy and viral suppression. Basically there's these exhaustion markers on the outside of the immune cell, and we kind of think of them like handbrakes pulled on the immune cell. So if you think of, you're sitting in your car driving down the highway with your handbrake pulled, you're never going to reach your full speed that you want to. Your car's going to be working a lot harder to try to get there. So basically that's what's happening in this immune exhaustion phenomenon in people living with HIV. There's these exhaustion markers, the LAG3, PD1, TIGIT, CTLA4, all these acronyms. They're these handbrakes pulled on the immune system cells. So that's a problem that we're facing right now in chronic HIV infection. Second part of my background is the type of immune cell that I study. So I study invariant natural killer T cells. So iNKT cells, they're a very powerful immune cell. They work more upstream. They link the innate and adaptive immune systems. They create produce a lot of cytokines, and they proliferate really quickly. And they kind of are an early domino in HIV response and response to other viral infections. So basically, you can see down here. Here's an iNKT cell. It's being activated by an antigen-presenting cell. This antigen-presenting cell is presenting their glycolipid antigens. They can be exogenous or endogenous. They basically stimulate these iNKT cells through presentation of this antigen. And then this iNKT cell produces cytokines, so messenger molecules, as well as proliferates rapidly. And you'll see this thing called alpha-gal SARE that I talk about in my assays. That's a gold standard in vitro stimulant. It's something we use in the lab to stimulate iNKT cells. It works this mechanism. So if you put the two of these together, in chronic HIV infection, we see that iNKT cells are exhausted and dysfunctional. So different studies in our lab included has seen an exhaustion marker called LAG3 increased on iNKT cells and correlated with decreased function. So if you look down here, just on the right for time's sake, on the x-axis, the more amount of LAG3 present on these cells, on iNKT cells, negatively correlates with the amount of interferon gamma that they can produce. Other groups have studied PD1, for example, and shown decreased iNKT cell function. Problem here, some gaps, is that there are very limited studies looking at exhaustion in iNKT cells in terms of there's lots of other markers to study, as well as the cohort demographics. Again, with only a few studies, there's lots of variability in the different cohorts that need to be studied as well. And also an issue with iNKT cells is that there's variable reports of being able to detect them ex vivo because there's a small group of the peripheral blood immune cells floating around in the blood. That's an issue. How I'm trying to address this, I won't talk about all the aims of my project, but a couple of them today. So one of the questions is, what is the functional and exhaustion profile ex vivo of these iNKT cells from people living with HIV who are virally suppressed on therapy art? So this is a cohort that I study. And then the second one is, so ex vivo is just right out of the gate, what do they look like? And then the second question I'll talk about today is the functional aspect of these cells. So when you actually put them to the test and you stimulate them, to what capacity do these iNKT cells from this population, how well is their function in a 10-day iNKT cell proliferation assay? There's some other questions that I do study as well. I won't touch on them much today. I won't show any data, but basically we also look at existing iNKT cell immune exhaustion with these things called immune checkpoint inhibitors. So they're very coming up in the cancer field and also moving to the HIV field, basically trying to release those handbrakes that are pulled on the cells to try to restore function. So that's something that I'm also studying. I'll just stick to the first two questions for today. So I have a total of 42 people in my cohort. I have some preliminary results to show you because we're not finished processing them all, but I'll show you an N of 7 from samples from people living with HIV and then also 5 controls. So basically what happens is we collect their blood, we isolate some of their white blood cells that are in the blood, and then we thaw them out and we rest them overnight, and then we do some ex vivo analysis via flow cytometry. So we look at markers of activation listed here, exhaustion, so PD-1 lag-3, but also TIGIT CTLA-4, and then senescence, which is kind of cell aging marker. And then I also did a 10-day alpha-gal ser iNKT cell proliferation assay in media with R10 and IL-2. And then we looked at, again, activation markers on these cells and exhaustion and proliferation. Were these cells able to respond and proliferate over 10 days? So first, sticking to the ex vivo analysis. So I just wanted to show you an example of flow cytometry gating. So here, for those of you who aren't familiar with flow cytometry, all these little blue dots on the graph here represent one cell that's taken up and processed through our machine. So basically we use different fluorescent tags to look at different markers on the cell. So here I have 2 markers that my cells are positive for, so we call them double positive, and that's how I find my iNKT cells. Then I move into those and I look at the expression of these activation, exhaustion, etc., all those markers. So just to simplify this, the x-axis of every thing here is the same. It's just the size of the cell and its density. And then the y-axis is changing. So we're looking at things like chronic activation markers here, and CCR5 expression, senescence markers. And then on the bottom here, this is how we identify who's positive for those exhaustion markers. And then we gate on them, and those are those gates there. So when we look at and we analyze all of our individuals, N of 7 here, the green group is samples from people living with HIV or iNKT cells, and the control is blue. So in terms of in B here, we're looking at activation markers. We see we're doing some non-parametric testing on the small sample size, some increased HLA-DR, some trending to CD38, and then also very high but consistent CCR5 expression. So these are some chronic activation markers. And then we move to exhaustion markers. We have some high PD-1, but that's not different than the control group expected. But we do see increased lag-3, TIGIT, and CTLA-4 in the samples from people living with HIV. And we also see increased senescence as well through the CD57 marker. So these are percent parent, so the percentage of cells that are expressing these markers. Should have said that earlier. And then after 10 days of proliferation, first on the left here, we see how many cells actually, or how many of these samples were able to actually respond and proliferate over the 10 days. We see their iNKT cell population after 10 days. So in 6 of the 7 individuals, we could actually see their cells. They were able to proliferate. We calculated this by fold change. They weren't able to proliferate necessarily as well as the control group, but we were able to detect them, which actually is a pretty big win. And then when we look here, we have activation markers after that 10-day readout. There's increased CD69, which is kind of an acute activation marker. So it looks like from people, samples from people living with HIV, that these cells are being very activated, and they're probably working very hard to become activated, but they do have increased amounts of exhaustion markers. In particular here, after 10 days, we saw TIGIT was significantly increased. So to kind of sum up everything, these iNKT cells were able to be detected in all 7 individuals, but they did show some markers of dysfunction, like chronic activation markers, as well as exhaustion markers. And then after the 10-day assay, 6 of the 7 populations were responsive, but we did see some elevated state of activation and exhaustion potentially concurrently at the same time. There's lots more to do with that data. I know there's so many other things I could show you. And then in terms of take-home messages, so basically looking at the applications of studying iNKT cells and immune exhaustion, we can use an HIV infection, and we can use this as a tool to assess functional status, identify biomarkers and correlates of disease in people living with HIV, but also looking at some interventional studies like those immune checkpoint inhibitors that I talked about, didn't touch on much here. And future directions, I do have to process more samples, so hopefully I can validate those findings. Just wanted to sum it up. I know I'm in the last minute here. How does this fit into the TB field and HIV-TB co-infection? Though it's not exactly what I'm studying, it's great to step outside of your rabbit hole you've fallen down in your grad school and pull yourself out. And for example, looking at iNKT cells in TB infection and then HIV-TB co-infection. There's lots of studies actually showing that iNKT cells are very important in control of TB infection and what happens in HIV when maybe those iNKT cells are depleted or dysfunctional. That could have some deleterious effects. Looking at the phenomenon of immune exhaustion in general in TB and HIV co-infection. So this immune exhaustion also happens in the TB setting, and you see some dysfunctional T cell responses and increased things like PD-1, for example. And then finally, talking about those immune checkpoint inhibitors, trying to remove those handbrakes off of the cells. There's some important findings on that because you can't ignore someone's TB status, for example, someone with cancer, but also with TB. If they take these immune checkpoint inhibitors when they have cancer to try to treat their cancer, there's actually effects of activating some TB, and there's also some interesting findings in HIV and some hypotheses as well that should be considered there too. But I'll leave that for the sake of time, but just some food for thought. So thank you so much for listening to me. I apologize I wasn't there in person. Thank you very much to the study participants, all the blood donors, my lab members, all my collaborators, and I'll take questions